in a eukaryotic cell, or for that matter in a prokaryotic cell also, the genetic information is present in the cell in the form of DNA. In the prokaryotes it is DNA, in the eukaryotes it is basically DNA and RNA. Now the eukaryotic system is a bit more evolved, and the genetic information is protected, it is kept inside the nucleus, whereas in the prokaryotes the genetic information is just scattered in the cytoplasm, it does not have a defined nucleus. So in the eukaryotic system there is a nucleus, inside the nucleus there is nucleosome, and inside the nucleosome the genetic material is safe and is protected from any damage. Now chromatin fibers are basically a thread-like structure which is made up of, or which contains DNA, RNA, basically the sugars, and proteins. Now these proteins are the histone proteins, and these histone proteins are also known as DNA binding protein. These DNA binding protein help the DNA to be tied up, okay, and there are 4 subunits of histone and there are 2 sets, so basically it is like an octamer, and that's why it's written there as DNA binding protein, that is an octamer. The DNA is known to carry a huge amount of information, and this DNA just cannot be scattered, it needs to be wrapped around tightly, and hence the histone proteins come very useful here. The eukaryotic DNA carries immense information, okay, it's a huge information, it's a huge data. Now how do you think that this amount of data is accommodated into a small space like a nucleus, into the nucleosome? Hence it's a mystery, or I would say it's really magical that this amount of information is there packed compactly, and that is only because of the histone proteins. Normally in the households we see the cable wires or the pipe that we use for gardening, the water pipe, it's all wound up, or it's coiled, and then it is hung on a nail, or it is coiled and it is stored in a smaller place. That's because anything that is much longer will consume a lot of space, but when it is coiled it can be accommodated, it can become compact and accommodated in a smaller space. So similarly here also the DNA here is huge, the information is huge, the chromatin fibers are in the form of string beaded structure. Now the only way to fit them is by tightly wrapping and coiling the DNA, and that's amazing. It's known, and scientists have discovered that they have observed that it appears like a string beaded structure under the microscope, but it is not so easy to see these structures, also because they are very thin and thread-like structures. So in a resting cell it is very difficult to spot chromatin. Chromatin fibers are in the nucleosomes of the resting cell. We know that the cell rests and then the cell divides, so it's not a constant process that the cell keeps on dividing, it's only when it is necessary. So there are times when the cell is resting, and when it is resting the DNA is there in the nucleosomes in the form of chromatin fiber. There are 2 types of chromatins, or you can say there are 2 forms of chromatin. One is the euchromatin and one is the heterochromatin. The euchromatin is a less condensed form, whereas the heterochromatin is a highly condensed form. During the cell division, that is in the prophase, the chromatin starts to condense, and this condensed chromatin is now called as chromosome. Now as they become condensed, the morphology changes. The chromatin are now converting into chromosomes because they condensed. Because of the condensation they become thick rod-like structures, and now these rod-like structures can be clearly seen under the microscope. From chromatin to chromosome, chromatin condenses to form chromosome. Now this chromosome duplicates, it forms an exact copy of itself. Now there are 2 chromosomes, and now these are called as sister chromatids because they are identical, the gene sequence is exactly the same. We are very well aware with the cell division via mitosis and meiosis. Mitotic cell division is an incremental cell division. A mother cell gives birth to 2 daughter cells. Now when the daughter cells are born, the daughter cells must have the material, the content. What will be the content there? it's going to be the cytoplasm, it is going to be the genetic material. The reason why the duplication of chromosomes happened is because when the daughter cells are formed, the genetic material from the mother has to be transferred to the daughter cells. So if there is less genetic material, then the daughter cells would not get the material. So hence the duplication happens because the genetic material is then received by the daughter cells. So the daughter cells just cannot be empty. They will require the genetic material from the mother cell, and hence the chromosome duplicates and the material is passed on to the daughter cells. So how do the sister chromatids exist? They are sister chromatids. The plural chromatids tells you that there are two of them, and these two are not separable. They are linked with each other. Just imagine a pair of scissors. The connection between the two blades, or the intersection point of the two blades, okay, something like that. So there is a constricted region, and that region is now called as a centromere. So basically the centromere is a point at which the sister chromatids are linked with each other. They form like a X, alphabet X. They look like an alphabet X. The intersection point is the centromere here,